Now students, in today's practical lab, we are going to study that how to determine ESR. So learning outcomes of the lecture are what is ESR, how to check ESR. We know that ESR is actually the erythrocyte sedimentation rate. It is the rate at which the erythrocytes settle down in a sample of blood that has been placed in a thin, tall, vertical tube. So this ESR is basically an important hematological test. It is used for the detection of inflammations by the infection, cancers, and autoimmune diseases. So the method which we are going to use in today's lecture is the Westergen's method. So for this experiment, we will need 1.6 ml of the venous blood, 0.4 ml of the 3.8% sodium citrate. You will need a pipette, sucker, timer, that is a watch, Westergen tube, and Westergen's stand. So the apparatus basically consists of a tube, that is Westergen tube, that is open from the both ends. The length is from 0 to 200 mm and the internal diameter is 2 mm. So we will need a stand that will be used to hold the tube vertically. So this is the tube and this is the stand for the tube. Now for this experiment, clean your antecubital area. This is the antecubital area with the help of an alcohol swab. Use a tourniquet and you will place or you will tie the tourniquet from 3 to 4 inches above the needle insertion point. So now after this, your veins will be more visible. Now, puncture the vein, draw 1.6 ml of the venous blood. Introduce this blood or add this blood into the test tube containing the anticoagulant, that is 3.8% sodium citrate. Now, this tube is containing 0.4 ml of the sodium citrate with 1.6 ml of the venous blood. Now, gently mix the tube in order to mix both solutions, that are blood and the anticoagulant. Now, what you will take? You will take a pipette. You will take the Westergen tube and you will attach a sucker on the tube. This is the tube. You will attach the sucker on the tube. Now you will start pressing the sucker. Now what it will do, it will suck the blood with the solution. It will suck the solution of the blood and the anticoagulant into the Westergen tube. Now fill the tube up to the mark 0. Now remove the sucker. Keep your finger at the tip of the tube in order to hold the solution in the tube. Now, fix this tube into the stand. This above clear portion will be read after 1 hour. After 1 hour, take the reading in mm of the clear plasma. This is the clear plasma which is above the settled RBCs. Now, this figure is actually representing this clear plasma. This, you will note down the reading of this clear plasma on the table, and this is actually your ESR rate. This is your ESR rate which is represented in mm per hour.